So welcome. My name is Clare Bryant. I'm Professor of Innate Immunity based in Departments of Veterinary Medicine and Medicine. And I'm here today with my colleague, Lalitha S, who will introduce herself when we get to it. I do first of all have to say that, the slides haven't changed, never mind. I do have to say first of all that our colleague Brian Ferguson can't make it today. Ironically, he has a virus. From a source that I won't be talking about called children. But I'll cover his slides. He and I work closely together. In fact, the three of us work closely together because we are from the, as well as our departments, we're from a newly established centre called the Centre for Pandemic Risk Management, which is a joint, which is led by two directors. I'm one of them, and the other director is Rob Doubleday from the Centre for Science and Policy. And it's a fusion of science and social sciences, particularly political sciences, policy sciences, working together to tackle some of the challenges in and around pandemics. And we're trying to do something really that's quite different from the other pandemic centres that are set up within the UK, working in combination with them, but tackling things that we particularly feel are unmet challenges within that. So it's very exciting. It's been founded by a philanthropic donation, and we will then aim to continue this, bring in further funding, and also develop a series of important outcomes, which will be things from policy documents through to apps, through to therapeutics, to really try and tackle future pandemics and infectious diseases in general. So with that in mind, the next pandemic, or the title of this session, is we will talk about three different topics. So firstly, where might it come from, accepting I'm not going to talk about children. Secondly, how might we manage it, which is what Lalitha will talk to you about. And then Brian would have talked to you about how can we intervene, but you'll get me doing that instead. So one of the things I haven't said is that I am a veterinarian. So I think a lot about infectious diseases in the context of animals, but also in the context of diseases that travel from animals to human. And so hence the reason I'm based in medicine and veterinary medicine. So one of the commonest routes for pandemic-related diseases is a zoonotic infection. So zoonotic infection is one where a pathogen, and so that's things like a bacterium, a virus, or a parasite, or a prion, moves from a non-human animal, and that's usually a vertebrate, into a human. But it not only has to move to humans, it then also has to transmit between humans. And that's the sort of definition of a zoonotic pathogen. Often these pathogens will have an intermediary species that can carry the pathogen, and often the intermediary species doesn't get sick. There are some exceptions. So for example, with COVID, you'll be familiar perhaps with the bat being the primary species, the pangolin, or in fact a raccoon dog now is probably thought to be the intermediary species before it came into humans. Hopefully you'll all be familiar with the global problems of bird flu, but I don't know if you're also familiar with the fact that bird flu has spilt over into cows in America. It's concentrating in the mammary glands of cows and is now spreading through milk. And this is now transmitting into cats, but also into humans. We haven't got human-to-human transmission yet, but it's a pretty alarming situation. So bird flu is another great example of a potentially zoonotic pathogen. And zoonotic infections really are of critical importance to humans because the last 5 epidemics have been zoonoses. So obviously COVID-19, but MERS, Ebola, swine flu, and the original SARS infection were all zoonotic pathogens. And I can give you other examples like TB, AIDS, various other things like that. These are predominantly they're all zoonotic pathogens, so there's a whole host of them. And in the pan-epidemic arena, the global cost to human life has been enormous. So COVID-19 estimated at 6.83 million, HIV 36 million people, swine flu was 12,500, Ebola 11,300, although that has changed because there's another outbreak of Ebola in Africa at the moment. And as I've said, avian flu is rampant at the moment, spilling over into cows, cats and humans. How does spread occur? Well, it can be vector-borne, by which I mean things like insects. Okay, you'll be familiar with mosquitoes transmitting malaria. Mosquitoes can transmit all sorts of diseases. The kind of things we can think about a number of viral diseases that spread from animals to people through a vector-borne disease. There's direct contact, which is where the human comes in contact with bodily fluids. So a dog that has rabies, if you come in contact with the saliva, you can get rabies from them, just for example. You can have indirect control, which is where you as a human come into contact with something where animals have contaminated a surface, where animals live or roam, and therefore you pick up a pathogen for that. And that's quite a common route for bacteria, for example, can spread that way. Foodborne pathogens, they're the things I think about a lot because I work on salmonella. So this is where a pathogen contaminates food. And then you can have waterborne viruses as well, or viruses and pathogens, so whereby you drink contaminated drinking water. And of course, many of the tropical diseases are transmitted in this way. So the example, of course, we understand best of all is the coronaviruses. So we really shouldn't have been surprised that we had the SARS-CoV-2 pandemic. Coronaviruses have been causing a number of pandemics, MERS and SARS-1, for example. But I think we chose the other way, really, and thought a lot about flu, which we expected to be the next pandemic, and were caught napping, really, because we should have expected coronaviruses to come along. And the real problem with coronaviruses, of course, was they were transmitted, or SARS-CoV-2 anyway, was transmitted from people who didn't show symptoms. So flu is transmitted from somebody who is symptomatic, but COVID-19 wasn't, and that was a major, major problem and the real challenge of trying to control it. So coronaviruses, for example, are common in bats, they're also common in birds. There are a number of intermediary hosts, pangolin, raccoons. This is a camel, they carry MERS, rodents. And what happened is you have to have an evolutionary process in the virus to allow it to travel into people. So the infamous spike protein, which you all have heard of, it was the adaptions in the spike protein that allowed it to change and mutate. In doing that, it was able to infect human cells because it could bind to relevant receptors, so ACE2, for example. Once it could bind to those receptors, it was internalized into the body of humans and then could grow and spread. And of course, as you'll all be familiar with, as the strains developed and mutated, and there's a nasty one about at the moment, they got increased transmissibility, and they had the ability to evade the immune system. So that's a really classic example of a very efficient zoonotic pathogen. Flu is interesting. So flu is in some ways slightly atypical. So it can infect all sorts of birds. But waterfowl, things like ducks and geese, they can carry the flu without actually getting much in the way of symptoms. In contrast, animals like chickens are actually very susceptible to bird flu, so they can actually get very serious infections and die, and it's a real welfare issue, actually. And so there's kind of already subtle differences between the strains and different strains of birds. But they can then of course, spill over into humans, either humans who are in very close contact with the birds, so this is where the farming industries become a problem, and why you should never pick up the carcasses of dead birds that you find out and about, because wild birds carry this as much as farm birds. But of course, now we know that for the H5N1 virus, which is the strain we're dealing with at the moment, the prime intermediary species of choice is the cow. And I have to say, this is a problem in the States. People are keeping very quiet about it. And it's not just due to Trump. I could easily blame Trump, but it's not actually. It's been going on for quite a while. And it's pretty rampant in the dairy herds in America. And the dairy industry are choosing to try and keep this quiet for reasons that I don't really understand. And as such, it has spilled over into people. There have been human cases. I think 2 or 3 people have died from it so far. But it's not actually making the transmission from person to person. And the reason why this is particularly frustrating is because there is a vaccine available. Okay, so we could easily vaccinate farmhands and stop the disease in its tracks. However, the American government have chosen not to do that. So zoonotic infections are increasing, and why is this? Well, of course, we've lived with domestic animals for millennia, and also with wild animals as well. But of course, there are a whole diversity of anthropogenic factors which have really intensified the animal-human interface. So this means that we're increasing our interactions with animals, and consequently the risk of disease spillover. So, and of course, this is driven by human population growth and the understandable efforts that we have to make to alleviate the associated poverty with the increasing population growth. So intensified farming, unsustainable exploitation of natural resources, culinary traditions, so wildlife meat consumption, or traditional medicine driving the trade of wild animals. All of these factors and many others can lead to potential spillovers. And of course, the problem for us all is that in an increasingly globalized planet, a spillover event that results in an efficient and sustainable transmission between people can then spread across the globe very quickly. And as I say, we've all seen that with COVID. So I've alluded to the fact that it's not just zoonotic infections that can cause potential pandemics. One of the real challenges, I think, and something I'm sure we'll all have thought about, and has been covered by many, many television dramas, of course, is the potential of the next pandemic to come from an engineered source. So what do we mean by that? And it's important to understand this because an engineered pandemic is quite an emotive term, particularly for me as a scientist. You know, the concept that you might think I'm in my lab making something to try and kill people is not an attractive concept, and of course that's not what we're doing at all. But what we do mean is a deliberate release of a pathogen for a variety of reasons, why anybody might think that's a good idea. Of course I don't understand that. But and we're exploring, and this is one of the things that our centre is doing, is thinking about deliberate release of a pathogen and what we might do, because it's an important thing for us to consider. We're thinking about the possibility that you could engineer a pathogen. So there's a lot of concern, for example, around artificial intelligence, and can you use artificial intelligence to engineer a pathogen to make it more nasty, okay, and more virulent? All of those things are theoretically possible, but to be honest, pathogens are really good at killing people. They don't really need any engineering. They're extremely good at causing damage. We're going to have to be very clever to make it be even worse. I think it's extremely unlikely, and I don't think that's the likely source of an engineered pandemic. But the potential for somebody to deliberately release something is something that we cannot exclude. And one of the other things that we also have to think about is this might not just be a pathogen that damages people. It's just as efficient to release a pathogen that damages animals, because that will strangulate the food chain. Even more efficient would be to release something that kills plants, because that would damage both animals and humans and strangulate the food chain. So in the work that we're thinking about in our centre, we're thinking very carefully not just about human pathogens, but about animal pathogens, which is probably not surprising to you as I'm a vet. But also we're thinking about plant pathogens, because potential pandemic can come from different sources and it can have different consequences. And of course, we can't rule out an accidental release of a pathogen. And indeed, for example, an example of an accidental release of a pathogen was when there was a leak at Clare Bryant laboratory. So that's the big animal virus centre in Surrey. They had an accidental release of foot and mouth disease, and we had a very contained epidemic of foot and mouth in and around the region of Surrey where the laboratory was. So these things do happen despite the best of our intentions. And so within the centre, we've been working with some really interesting people, including Lalitha and Rob. And we had a visiting scientist from the States who worked a lot in warfare scenarios, and we were talking about how might we can think about the management of a pandemic in the immediate term, and what might we do in the long term, because we need to be able to have strategies that are ready to go at any time, which involves a whole different way of thinking to the kind of frontline battle that we saw during COVID. And so he used a couple of terminologies that people think about when they're thinking about strategies for, you know, when you're at war. And so he said there's a theory of a victory in a pandemic. So these are the kind of immediate things we do, the sort of public health strategies, the politics and the science. And having a vision of what the end of the pandemic looks like. And I'm not sure we had that in COVID, okay? It kind of just dribbling along now. And I'm not sure that's really what we wanted. But there's there's a whole bunch of discussion we could have in and around what we would do in the face of a pandemic. But also, just as important, is a grand strategy or a theory of population health security. And so this is the way in which we have to work with the national leadership to continuously work to produce a durable health security for the population. So this involves investments in different directions to think about vaccine strategies, manufacturing strategies, PPE strategies, public health strategies, surveillance strategies, a whole panel of things that we need to think about. And so this is kind of what we're trying to do, which is couple this kind of strategic thinking with our science to move forward. And on that note, I'm going to give you to Lalitha.
Thank you very much, Claire. So while Claire makes her way back to her seat, I'll introduce myself. I am a senior research associate at the university. Like Claire, I've got also many different affiliations, maybe not quite as many. I work within the Center for Pandemic Risk Management with Claire, and I've been doing so for a few months now. But I also have an affiliation to the Center for the Study of Existential Risk. And I think that you'll be able to see the crossover in some of the topics that I'll talk about. My background is as a parasitologist, but more recently I've been getting more into biosecurity, including things like the harmful use of disease, biological weapons, and counterproliferation, and how the lessons that we have learned from that may be applied to being ready for the next pandemic, wherever it may come from. So as Claire mentioned, we have come across the idea of an engineered pandemic, or certainly the phrase engineered pandemic, a lot recently in TV, in movies, but also in headlines and also in conspiracy theories. And it's quite an evocative, emotive phrase, and it conjures up these images of mad scientists or Bond villains in their underground lairs unleashing the next whatever it is onto an unsuspecting population. But as researchers, it's our job to actually ground this phrase and to unpick it a little bit, to parse it, to see, well, what are we meaning when we say an engineered pandemic? What are others meaning when they say engineered pandemic? And can we realistically and plausibly assess the different elements of that in order to research things that might be useful for what we may be facing in the future? An engineered pandemic implies an engineer of some kind. So we've got to start with who. Who might engineer what? How might they engineer it? And for what purpose? And each of these can be dissected into its different elements. The who, we have some examples in history of many states, over 20, having had state biological weapons programs, including the US, including the UK, including Canada, including several others. Since 1972, this year marks the 50th anniversary, we also have a biological weapons convention which bans the use of the life sciences for weaponized purposes. But that doesn't necessarily preclude it. People do, countries and states do things maybe covertly that contravene conventions all the time, unfortunately. So we've always got to be prepared for that to happen again. But it might be some sort of a state biological weapons program either deliberately or accidentally unleashing some sort of a pandemic event. Especially since 2001, since 9/11, there's also been increased concern about so-called non-state actors who have, to a greater or lesser extent, expressed interest in the use of the life sciences for harmful purposes. And again, you've got to consider the possibilities that these kinds of actors may be seeking to misuse biology in the pandemic space deliberately, but they also might do so accidentally on the way to achieving other aims. And also, there is the possibility of legitimate scientists doing really public-minded research, but having a lapse in safety or security and containment, and therefore releasing something that oughtn't be released. And again, the narrative is most often that an engineered pandemic is the pathogen being engineered. So let's consider that for a second. What is it about the pathogen that one might want to engineer? Its transmissibility, its lethality, the kinds of species that it can affect, or how long it takes between infection and the exhibition of symptoms, any of those elements. And I think in popular narratives, there's this misconception that each of those characteristics is like a knob that you can turn from 0 to 10, and that with AI, your malicious actor might just turn them all up to 10, and that would be that. It would be very easy to do that. That is not how biology works. There are strong evolutionary processes at work that constrain these, and we're not exactly sure how that happens yet, which is all very good for us because it means that engineering a pathogen is very difficult. But as Claire mentioned earlier, you may not need to because our natural pathogens are quite bad in and of themselves. But now, apart from the pathogen itself, I would like you to consider other elements of the world we live in that could be engineered, that would and already have made pandemics either pandemics when they could have just been outbreaks, or that might make pandemics worse. And that could be things like engineering the social, and the political, and the built environment. And an example of this is the emergence of HIV in Congo. At the time, it was a center of colonial power, and there was one or more crossover events that meant that HIV emerged into the human population. And most likely, it would have stayed there in relatively small localized outbreaks, were it not for the fact that it was a colonial railway center, which meant that lots of people were coming to Kinshasa for work, including sex work, and it was a railway hub, which means that those people were traveling elsewhere as well. And so it was that engineering very literally in terms of the railroads, but also engineering of human populations moving all over the place, that meant that it became eventually a pandemic. The same with COVID. If we didn't have airplanes, COVID probably wouldn't have been a pandemic. But of course, that's the world that we've engineered around us to do all the things that we enjoy doing. Slightly on a different sort of plane, but still engineering, I would argue, is the engineering of the information environment. I alluded very briefly to conspiracy theories, but we're seeing so many examples of mis and disinformation around health, around vaccines, around the source of COVID and other diseases. And that is an engineering, sometimes very deliberate, of the information environment that will ultimately prevent us from doing all we can to stop the next pandemic in its tracks. And this is closely related to an engineering of the governance environment as well. Quite a lot of the misinformation and disinformation that we've seen over the past few years has been accusations of one country or another developing biological weapons capacities, when that is absolutely not supported in fact. Why might an actor, in this case Russia, be attempting this in order to undermine the governance structures that we do have, that we rely on, like the Biological Weapons Convention? So by by reducing global trust in the multilateral institutions that we have, we hamper ourselves in trying to do that oversight and to prevent instances of these engineered biological uses that very unfortunately for us. So we've got to keep ensuring that our multilateral organizations are themselves strong to help us. And the how is quite related, because again, it could be an accidental release, it could be a biological weapons use or biological terror, but it could also be an exacerbation of a natural outbreak. And as Claire mentioned, we've got to think beyond just the humans, because there are ways that one could harm the animals and plants that we all rely on that could actually ultimately be more harmful and in a certain sense easier to engineer for a malicious actor. If you think about someone seeking to develop a biological weapon against humans, it's very difficult to test that. Whereas testing on a plant or testing on an animal to see whether your weapon works would be quite a lot easier. So you've got to consider that as well. The why question is where things get really quite interesting and quite sticky, and where being in an interdisciplinary center like ours actually affords us the opportunity to really delve into it. If we think about the kinds of actors, a legitimate researcher, we have to think about, well, why are they doing the research that they are doing? And what are the boundaries around safety and security that they're considering in their own work? But we can generally assume, we can assume that they are pursuing the research that they're pursuing for the ends of public health. But the malicious actor who's pursuing a biological weapons program or a biological terror event, what is the motivation? that's much more diffuse, that's much more diverse, and we don't have great empirical ways of understanding those because we are not in the intelligence service, for example. We are academics. However, there's something we can learn about past actors, past events, as well as current actors that we think might have explored the idea of misusing the life sciences for malicious purposes, and trying to understand things like what might motivate them, which would inform things like theory or doctrine, and therefore what is the utility of a biological weapon for their ends, and do those things align, do those things match? But also we can explore things like current levels and interaction with novel biotechnologies or existing biotechnologies, and things like AI. Are certain groups very likely to embrace and utilize new technologies in the development of the weapons that they use to further their aims? So it's very complex, and it involves social sciences, and it involves counterterrorism studies, it involves legal studies, and it's a really exciting space to work, trying to understand these deeply social and political elements, and then try and match them to the biological and the life sciences realm, and trying to unpick that and understand it. So already, with just the phrase engineered pandemics, you get a huge matrix of possible scenarios, and that's not counting the natural emergence of pandemics or the accidental emergence of pandemics in plants, in animals, in humans, in zoonotics. So how do we actually go about trying to capture this huge world of potential pandemics? One way is by asking, what do we prepare for already? Or more accurately, what do we rehearse for? And I use the word rehearse very consciously, because many organizations, including the UK government, governments all over the world, but also think tanks, universities, the WHO, WOA, the World Organization for Animal Health, run tabletops. These are scenario exercises that literally rehearse the emergence of a disease somewhere in the world, and what would we do? We an organization like WHO, or we an organization, a department like the Department of Health and Social Care, or we as the post office. How might the post office deal with a pandemic? Everybody rehearses this to a greater or lesser degree. So one of the first research projects that we've embarked on at the center is to try and do a meta-analysis of these exercises, to try and find out what are people rehearsing for. And we're doing this on a number of levels. The first is process. Not all tabletops are alike. First of all, annoyingly, they're not even all called tabletops. They're called tabletops, they're called war games, scenarios, or drills, simulations. There's a whole number of them. And they often have different purposes. They involve different sets of actors who bring different skill sets into them. They involve different organizations. One of the things that we're looking at very carefully is, what are the exercises for? And this is not always, at one level, they are to prepare for a pandemic. But what level are they actually exercising? Are they exercising things like, does this minister know who to contact at this other department? Which is very much an organizational question. Or are they asking questions like, do we have access to particular types of PPE? Which is a very different logistical question. So what we're assessing is whether the form of the exercise matches the function of the exercise, and whether it's clearly defined what it is that it is for. Next, we're also looking at whether these exercises themselves are assessed. And that answer varies widely. And then if they are assessed, what happens to the recommendations that are drawn from the exercises? It's no good if they just go and sit in a drawer until the next pandemic actually happens. So as well as process, we're also looking at content. And this is very much looking at the scenario that plays out in the exercise. Thinking about, when during the pandemic was the scenario placed? And actually most exercises are at emergence, which on the one hand is quite obvious, because that's when you first need to make difficult decisions about picking up signals from noise. How do you, set your machine in motion if you're not actually sure this is going to end up being a pandemic? But this also relates to difficulties in the process of how you actually run an exercise. If you're spending a lot of money, and these kinds of exercises can run into the millions of pounds or more, you're not going to really run an exercise and say, well, that was an outbreak that fizzled out. You've got to actually run a pandemic to have a successful pandemic exercise. But we're also, in this section of looking at the content, we're also looking at properties of disease agents that people are worried about and therefore exercising, but also things that are built into the exercise. Assumptions and discussions about things like what countermeasures are assumed to be available, like are we assuming something about vaccine development, about PPE, about hospitals that we may or may not be sure about at the start of the exercise, let alone in the start of a real-life pandemic. We're also looking at, within these exercise scenarios, how are assumptions about societal impacts and the actual world in which this fictional pandemic emerges, what are the assumptions built around those? And importantly, things about social capital and social authority. In the world of the exercise, if you institute a lockdown, what are the assumptions being made about whether that will be followed? Does the authority actually have the impact that it thinks it has? So we've gathered, you don't need to really look at the detail, but this is a snapshot of what we've been looking at. We've gathered over 50 exercises, and our criteria was that it had to be pandemic, but they have been run in our data set by a number of different organizations that I showed on the previous slide. We've concentrated a lot on the UK government ones. And this is color-coded by the type of exercise and the pathogen type. In blue, are the exercises that have involved flu. So to echo what Claire was saying before, we've done a whole lot of preparation for flu. That said, we have also, in purple, prepared for coronavirus, a not insignificant number of times. We've run exercises on coronaviruses. And then in gray are the ones that are other. These tend to be things like smallpox or plague, that they're known pathogens that we have done some exercises. And then there's also in bright green the pathogen X, which is really a pathogen that is not closely related to any known pathogen. However, when you delve deeper, a lot of those pathogen X's tend to have similar characteristics to flu. But we do nominally prepare for a pathogen X. And then the shapes of the dots are whether they are natural or biological terror related. And the vast majority that have been exercised are natural, although again, I think 7 of them have been non-natural causes. A slightly deeper look is we're doing a deeper look into ones from the UK and ones from the Johns Hopkins Centre for Health Security as really deep case studies. And this is just an example of the kinds of data that we've been extracting from exercise reports, and that will then aggregate to do a meta-analysis. And our very initial findings on this is that mainly what are exercised are human or zoonotic pandemics. There are very few panzootics, with the exception of avian flu that doesn't cross over into humans. And panzootics are pandemics that would circulate amongst animals, but that would not cross over into humans, which would have huge impacts in terms of our agricultural capacity, but they're not exercised in the same way. And we're investigating now that it might be that it's a question of terminology that we need to be careful of in our research methods. Very few accidents that have been modeled. In fact, I think there was only one accident that was an accident that was made by a terrorist group. So it may be that this is a gap in the way we exercise. No plant pandemics in any of the data set. Again, this might be an issue of terminology, or it might be that the temporal scale at which a plant pandemic would unfold is not something that lends itself well to be exercised in a tabletop in the same way that we do for these other types of pandemic. Again, active areas of investigation. And some trends that we can find are that the exercises tend to be performed in waves, following 9/11, following West African Ebola, following SARS-1, and following COVID, of course, as well. And so the emergence or the fear of a real event sparks a whole wave of preparing for that event once it's happened, which I think is not unfamiliar to us. And I already mentioned the types of pathogens that have been used. And unsurprisingly, perhaps, recent exercises emphasize international coordination, especially around things like vaccine and equitable access around that. And again, reflecting the lessons from the very most recent pandemic. Which begs the question, are we always preparing for the last pandemic? But maybe that's okay. Flu, despite all of our preparation, is still an incredibly likely pandemic to happen. And it's right that we should keep preparing for it. So maybe that's okay. It's not always necessary that we look for the very strange event, but we should spend a little bit of time looking for that as well. Now I'll hand back to Claire.
Okay, so were Brian here, who is a very experienced virologist, he was going to talk to you about interventions, because by now I'm hoping you're feeling uncomfortable and thinking, oh dear, what are we going to do? So, of course, some things have moved on at a massive pace. So vaccines, of course, vaccines was a win in COVID, and we'll come back to talk about this. Antivirals not, okay? And there are various other things we had during the pandemic, including things like monoclonal antibodies, which are antibody ways of trying to interfere with viruses. And these are all sort of pharmaceutical interventions. One of the common features, though, within the pandemic is that if you don't actually manage to stop the virus, what people die of is a condition called sepsis, which is where the whole body becomes super inflamed and shuts down. And I've put that in brackets because we have nothing to treat sepsis, and that hasn't changed since the pandemic, I'm afraid. And it's something I'm very interested in. But it's a huge problem because it's a very difficult disease syndrome to target, and lots of pharmaceutical companies won't touch it. So that remains in brackets as a problem. Of course, we had a bunch of non-pharmaceutical interventions. So, of course, you'll all remember social distancing, you'll all remember face masks, isolation, quarantine. And of course, we had public health measures. So a big win in the pandemic was surveillance. And we're very, very good at surveillance. Of course, we've racked that all back, and we're not doing anywhere near as much as we used to, which is kind of a pity, but we are very good at it. We weren't great at contact tracing, but I think that would be a bit of a different deal now, and I think that would be managed completely differently, especially with the rise in tech, which is completely changing that sort of problem. Travel restrictions and border controls, that could have been done better in the pandemic, as I'm sure you'll remember. And I think one of the things that we really learned from the pandemic was how heavily stressed our healthcare system and our fantastic healthcare workers were. And the NHS and dealing with and trying to help the people that helped us during the pandemic is an ongoing problem. And it's something of complete concern. How do you support people? I had a respiratory physician in my lab, for example, and I would talk to him and he would say, well, we don't know what to do. We have no idea how to treat people. We just don't know what to do. And when you've been trained as a medical vet, then you're trained to, you know what to do, you know how to treat it, and you therefore follow a program. And this has been massive. People who had to deal with this, some people are an equivalent to post-traumatic stress syndromes because they've been dealing with these kind of problems. So the healthcare system management needs a lot of thought. And then public communication and engagement is also an interesting facet. I don't know how you felt we did as public engagement people during the pandemic. I did a lot of it. Thank you for that vote of confidence. I would agree. So, and I think I felt very much by the time I started to get involved around the time Omicron came along. And I spent a lot of my time telling people on the radio and television to get vaccinated and trying to explain how vaccines worked and so forth. But we'd kind of already lost the battle by this point because I also did a number of radio phone-ins, and the number of really bizarre questions we were getting were absolutely clear that social media had got a grip of a certain proportion of the population, and it really didn't matter what I say or did, it wasn't going to make any difference. So I think we've got a whole question around social media and the failure, the lack of trust I feel there now is with science and medicine. That is a problem, and I don't know how we win that battle back. So vaccines, everything has changed. We've got whole new classes of vaccines. Okay, the mRNA vaccines are a revelation. Now, they're actually something that's been in the pipeline for a long time. They're being developed to treat cancer. So what probably wasn't really understood was that a lot of the basic biology was sorted out, and in fact, they were about to go into clinical trials for cancer. The guys working on it reorientated it and said, we're going to develop vaccines for COVID and see if it works, and it did. And it was remarkable because we had very, very quick vaccines. But then a question that I got asked, actually, this on the next slide, I think, was it not there, maybe? A question I got asked a lot was, were the vaccines safe? Because were there lots of jumps made in how we did things? And the answer was no, there were no jumps made in anything that we did, in fact. Because what normally happens is there's a long, rambling pathway to get to vaccine approval. And you have to go through a series of steps. In COVID, what happened was that everybody worked together, from the regulators to the basic scientists, and there was a lot of money thrown at it, so that all the normal steps that would have taken years were done more or less in parallel. So that the same process was gone through, but the money and the urgency meant that we were able to do that work in a logical, rapid time frame, which then asks a whole new set of questions, which is, right, okay, what can we learn from this, and how can we make our pipelines much more efficient to get things like vaccines ready and waiting for us to use in the future? So I think that's a whole interesting question to ask. We are missing a lot of vaccines for a lot of pathogens. You'll know that. We don't have an HIV vaccine. We don't have a dengue vaccine. TB is still a problem. Malaria, there's some vaccines we're waiting to see. There's an avian influenza vaccine now, just about. And of course, as soon as a new disease comes along, then all bets are off and we have to think again. So the way in which we think about this, of course, is trying to really do the research on understanding how the body responds to different viruses. We can use experimental approaches, because a lot of the research has been done on mice or animal models, and that doesn't necessarily recapitulate what happens in humans. So now we're at a stage where we can do experimental medicine trials, and we can do trials in people to really understand what's going on. And that's not with pathogen, but it's with bits of the pathogen which will stimulate a similar response, so you can understand what's happening. There's a lot of pathogen research and a lot of surveillance, not as much surveillance as we would like, but it's still ongoing. And of course, we have ways of increasing the speed of development. And importantly, thinking much more carefully, I think, about how we integrate what's happening with research and development with manufacture and distribution, because actually some of the real big problems during the pandemic was that we couldn't make enough of the stuff. We couldn't make enough PPE, we couldn't make enough ventilators, we couldn't make enough drugs, we couldn't make enough vaccines. So how do we manage the making bit more? And then obviously the other thing that was really awful during COVID was the fact that there was no equitable partnerships. Nothing. We had our vaccines, but we didn't care whether anybody else in the world had them. And in doing that, the classical example of why that's a massive problem was, I mentioned Omicron. So Omicron arose in South Africa. South Africa didn't have any vaccines. We had vaccines, the South Africans didn't. And a new strain arose. And then that spread through the world. That was mutated, and that caused us all sorts of problems. So, with a pandemic, you need a global equity to actually tackle a pandemic where it is, not just randomly vaccinate people who've got the money to get vaccinated. So there's a whole ethical discussion that needs some thought. This is the pipeline I was talking about, and effectively, the top is the traditional pathway of the rambling pathway of getting something approved, a vaccine approved. And this is what happened during COVID with the Oxford vaccines and the mRNA vaccines. We had enough money to make everything overlap seamlessly. So we went through all the right steps. But the seamless process and the innovative work actually done by Kate Bingham and her crew meant that we maybe manufactured what we needed at the time. So you can do this if you have lots of money. And we put a lot of money understandably into vaccine development during the pandemic. But we did everything the same that you would do for a normal vaccine and the normal vaccine safety pipeline. So we're doing a lot here. This is just a graph to show you the sort of things and the sort of areas that people work in. Brian forgot to put me on this, so I gave him a telling off, but I'm in this bit here. I thought, Brian, really? So we do everything from looking at the bits of the pathogen that you want to generate a specific response, right the way through the preclinical, clinical development, economics, clinical trials, policy implementation, all the way through to be able to roll this out. So we have a very broad group of people in Cambridge. And one of the things I think that COVID has done is made us think, okay, how do we knit all this together so that we can work much more efficiently as a cohesive whole? And I'm sure you're all alumni, so you'll probably be aware that Cambridge tends to work in a kind of distributed type way. I can't think how else to describe it. I've been here a long time. So, trying to join things together is not something we've traditionally done, but it is something that we are very much thinking about much more now than we used to. Antivirals, there are some success stories. For example, there's a new treatment for hepatitis C. This is an interesting treatment because it inhibits two parts of the virus pathway. It inhibits the construction of new viruses, and it also inhibits the replication of new viruses. And this is important because if you can target two different points in a pathogen's growth and distribution pathway, you reduce the opportunities of getting resistance. So the concern during COVID was that we made vaccines against one protein in the COVID virus, the spike protein. And then what the virus did was mutate to evade that, and we had to keep on changing the vaccine. Okay? So when you have a two-strike hit, you reduce the chances of resistance. So this drug now has been approved for use against hepatitis C and has a 95% cure rate, so that's a success. But it's kind of like the rare light in the dark world of antiviral development, because really we're not making enough progress. We haven't got antivirals, really any good antivirals anymore for influenza, mpox, dengue. So Tamiflu, you'll be familiar with Tamiflu. You remember we lived through the swine flu crisis, and we used Tamiflu then. But of course the problem was the virus started to develop resistance to Tamiflu, and timing of the use of this is also critical. You have to use it very quickly within the first 48 hours of flu symptoms. So there's a whole bunch of issues there as well. And we don't have any therapeutics against things like the cold virus, as you know, against dengue, Zika, mpox, Ebola, and disease X. So there's a whole bunch of virology research that's going on, virus surveillance. We're beginning to work with colleagues in AI to try and really develop new drugs and to work out how to decrease the pipeline of time it takes to identify them. And again, integrating research and development with manufacture and distribution. Central to this is making sure that we use combinations and repurposing to try and combat resistance, and I think that's something we have to be very aware of. So the summary here, to give you a chance to ask us some questions, is we're kind of at the point where we've got vaccines. I think that we're in good shape for vaccines. Brian thinks we're in good shape for antivirals. I don't think we are, because there's not a lot of will in big pharma actually to make antivirals. So my personal view is we need a little bit more thought about that. Sepsis, we have nothing, so it remains there in brackets. From a public health point of view, I think surveillance and contact tracing, certainly surveillance we're in good shape for that, but contact tracing not really. And then healthcare management and public engagement, well, we tried, but we could do better, would be my answer for that. And so what I'll leave you with then is our logo again, to say this is the Centre for Pandemic Risk Management, and we're from there. And what we're trying to do is to join all the bits up. Okay, we've given you lots of examples where actually if everybody was thinking together to try and come up with cross-cutting solutions, you would do it by working across disciplines. And this is, our centre is a genuine multidisciplinary science and policy centre to try and join all the bits up to develop a cohesive strategy and intervention strategy in order to help us fight the next pandemic. And we'd be very happy to take any questions. Thank you very much.
Thank you both for very stimulating talks. There's a factor to do with pandemics that you haven't mentioned except in terms of a general need for public education. And that is the horror of seeing lots of people you know falling ill. My father was in the generation who caught Spanish flu just at the end of the First World War. And I remember friends describing the HIV outbreaks in Africa, which were happening for many years before it reached Britain. I'm glad you mentioned our Oxford colleagues. Adrian Hill was my student 40-odd years ago, who developed the Oxford AstraZeneca vaccine. And I have a particular concern when I did one of those tabletop exercises with my MSc students in public health about a decade ago. So our knowledge of pandemics was more limited then, but my students, many of them came from the developing world, and they were convinced that if we have a disease X, it's already with us, just somewhere remote, the way HIV had been in the Belgian Congo. And we went round the table, and the lead candidate came from a friend from Chile. I bet you can guess what it was. Well, if I say that I had worked with hantaviruses in my youth, these are usually spread by rats peeing on human food. The old name was Korean hemorrhagic virus, if anyone is old enough to remember the Korean War. What I hadn't realized until my Chilean student explained it is that there are respiratory routes for being infected with a hemorrhagic fever like hantavirus. And the effects on people are very gruesome, heartbreaking to see, really. So although I agree that flu is the most likely candidate, and it spread so quickly, the collapse of morale that happened across whole military units at the end of the First War, whole towns. If you were in a remote town in Norway, Iceland, people just lost hope when they saw all their neighbors falling ill. However, in terms of the danger of spread, my disease X would be something like Korean hemorrhagic fever spread by a respiratory route. And I don't know if you have any thoughts on that, because it already exists. It's there in the Andes. It's just in remote communities.
Yeah, so I agree on both fronts, and we've talked a lot about mental health support, by which we don't just, not just for health workers, but also how do you, I mean, that was really when I was giving you the example of the respiratory physician. He was pretty much in a state of shock most of the time, as were most of his colleagues. And it was very, very hard to see. And I think the other thing I certainly saw a lot of in managing a group of younger people, dealing with people, either dealing with people dying or dealing with the consequences of being locked up or dealing with the consequences of sickness, was actually very difficult. And long COVID, there's a whole bunch of very important mental health issues in and around that, I think. And I agree. I don't have a solution for that. And it's not something that we're ignoring, but it's not something at the moment that we've got a programme on. But I agree, it's very important. With respect to hemorrhagic fevers, I also agree, right? Horrific, right? Horrific. And there's obviously a lot of work being done by Ebola, and in fact the Oxford unit have indeed developed a successful vaccine to help against that. I think possibly the difference between that and COVID, the thing that shook me the most was the inertness of the UK government in the face of COVID. I mean, it's just, you couldn't make it up, right? And I think we have to be thankful that Boris Johnson got really sick. I think otherwise we would have been like the Swedish experiment, but with a higher population density. And that, to me, that was the biggest challenge with COVID, was because it was asymptomatic. The hemorrhagic fevers cannot be loosely described as asymptomatic and also very severe. And I suspect that the response would be swifter and much more aggressive in the face of that. And this is one of the things that we're modelling at the moment. So we have one of the people who's working with us in the centre is Julia Golk, who's a very eminent mathematician who works on disease modelling. And she said, you know, Claire, the trouble is with the mathematical modellers at the moment, she said they were all on flu, she said they're now all on COVID, and it's like they're blind to anything else. And I said, well, Julia, we need to do other things. And so hemorrhagic fevers are one of the things that we'll be looking to model in the group. So yeah, I completely agree with you.
Hi, first of all, thank you both. Having done the meta-analyses on tabletop exercises, are they worthwhile? And if so, what do you actually learn that then gets implemented?
My get-out clause is maybe that the analysis is not done. We are just at the beginning of our analysis. And it's a really difficult question to ask, actually, because are they worthwhile for what purpose? Yeah. But for example, there are some tabletops whose purpose is to raise awareness. Oh, okay, fine. What does that actually mean? How do you measure that? Has it worked? Which is a very difficult question to answer. But if it's something like, do all the different bits of the system know how to contact each other? That's something that you can measure, and that you can say, yes, we are doing that well, or we're doing that sufficiently, or no, we aren't. And that's a very clear lesson that can be implemented. The question is, are they implemented? Yes, that is the million-dollar question. There are quite a few of these tabletops that are successful on the evaluation of participant surveys, which unfortunately most of the time are, I had a great time, let's do this again next year. I would argue from a public health perspective that's not all that useful. And I think what we're working towards now, and I've seen a couple of examples of this, is where you don't want the test of whether the exercise was successful. You don't want the test to be a pandemic. So we've got to figure out other tests that don't involve really the big test. And I think that it's a case of embedding the lesson incorporation into business as usual, because nobody's got the time to spend a week, a month, or a year just implementing lessons and then getting back to the day-to-day business of running the organization, the country, the company. You've got to figure out a way to make that part of the day-to-day activities. And I think there's an increased awareness that this needs to be done and an exploration of how this might be done. But I wouldn't say that it's 100% done yet. But I think just a slight thing is that I've seen quite a lot of really interesting tabletops, not quite a lot, more than I used to, that have incorporated things like mis and disinformation, which I think is a useful way to think about really what is an emerging phenomenon that we're not exactly sure of the dynamics. And these tabletops are often a way to investigate these phenomena. They're not always intended to provide lessons. They can be exploratory and imaginative and creative to look at the world of plausibility, as well as this is the list of 5 things we must do. That doesn't really answer your question, but I hope it was interesting.
Thank you. Thanks very much for a very interesting talk. Have you any ideas on how to control this phenomenon of activity, actually, of gain-of-function research? Bird flu, my contention was called bird flu for a very good reason. And then in 2012, I think it was, professor at the virology lab of Rotterdam University, Ron Fouchier, announced that he had succeeded in engineering the bird flu virus to be really infectious to humans. Quick on his heels was followed by Professor Kawaoka at Wisconsin University, announcing a similar thing. Most recently, you're probably well aware that a group in Korea announced that they had engineered the bird flu virus so that it caused a 40% mortality to humans, or to mammals, I should say, at that point. What is being done to really police and put a halt to these activities, which seem to be funded by the same organizations that will gain from so-called vaccines?
There's 2 prongs to that answer. So I'll answer a little, but I'll hand over to Lalitha, because it's something we're thinking about a lot. So for me, when I put in my grants, which I have to do it regularly, not so much now that I'm done, but anyway, my research is very tightly controlled by the UK government and by the funders. So gain-of-function research is something we are not allowed to do, and that is incredibly tightly regulated, both within the university, within the department, and within the country. So, Lalitha, do you want to take this on? Because this is something we've been thinking about. Globally, in America and places like that, it's supposed to be more tightly regulated. But you can see, as you described, examples where, it's not, and partly in the UK it's been tightly regulated because of what's happened in the past, right? But it's something we're anxious about, right?
Yeah, and I think there are several different prongs to this. One is the oversight of research that you think could actually have a beneficial outcome, but you've got to take some risks along the way that makes it worthwhile anyway. And that, I think, is a kind of research where you can say, okay, well, we'll govern it, we'll oversee it extremely carefully, but ultimately we think this is necessary research, which is quite different, I think, from exploratory, let's try and do it if we can, just because we can. And the trouble is that it's often difficult to know the difference. You know, someone may not be intending to perform a gain-of-function experiment, but it ends up looking like that. And so predicting these things in advance is not always that easy. So we've got to figure out mechanisms to do that. And I think it's got to be done at the level of funding rather than at the level of publication, which was the controversial, you know, which was the two cases that you described, Fouché and Kawaoka. It shouldn't have been up to the publishers. It shouldn't have been at the level of publication that the decision to pursue that research or not. That was far too late. And arguably, those cases have also been very counterproductive in plain old normal non-gain-of-function virology, where the backlash against those kinds of experiments have made it much more difficult to do conventional, very safe and very beneficial research. In the US, for example, there have been swinging cuts on all kinds of pathogen research, on TB, for example, which has nothing to do with gain of research because it's been an overreaction to 2 experiments, or 3 experiments. So I think it's a very difficult line to tread. And the final thing is that I would say it's interesting also in those 2 cases, but in the whole narrative around gain of function, is the concern around, we have to be better at articulating what is the concern in order to then better institute safeguards that would mitigate against that. Is the concern of escape from a laboratory? We have a certain set of measures that we can reinforce to guard against escape from a laboratory. Is it a case of we're worried that a malicious actor might read the paper and then replicate that research? That may also be a concern, but that's a different set of measures that we can work on and institute and strengthen. But just saying we're concerned, it's risky, we've got to dig a little bit deeper than that. No.
Okay, we're going to wrap up. Sorry, we're just running over now, and I know people have to get to their next session, so thank you.